Introduction, Dr. Watt. Dr. Kevin Watt received his MD and PhD degree from the University of North Carolina, Chapel Hill, and he completed his pediatric residency and pediatric critical care fellowship and research fellowship at Duke University. He was a faculty member at Duke for 7 years before moving to the University of Utah. He is the Robert M. Ward Endowed Chair in Clinical Pharmacology and an Associate Professor of Pediatrics at that institution. He also serves as the Chief of the Division of Clinical Pharmacology in the Department of Pediatrics and treats patients in the Pediatric Intensive Care Unit at Primary Children's Hospital. Dr. Watt's research career spans over 24 years at institutions such as the Thomas J. Watson Foundation, J.W. Jones Ecological Research Center, and the Duke Clinical Research Institute at Duke University. Today, he's going to he's going to speak about his leadership role and the opportunities attendant with the Collaborative Pediatric Clinical Research Network and the NCATS-funded Utah Trials Innovation Center and HEAL Initiative. Thank you, Dr. Watt.
Thanks, Rob, and thanks everyone for inviting. As Rob mentioned, I'm going to talk today about the Collaborative Pediatric Critical Care Research Network, or CAPCORN. Next slide, please. So CAPCORN was originally funded by NSCHD back in 2005, and we're currently in the 4th cycle. During the first 3 cycles, it was structured like a typical network with a pool of money to fund various studies during each of the cycles. And during those first 3 cycles, we completed 35 studies or trials, from pharmacology studies to some of the pivotal clinical trials in pediatric critical care. We've enrolled over 10,000 critically ill children during that time and have over 200 publications from this work. The current cycle, cycle 4, is structured a little bit differently in that we were funded to conduct one large multi-center trial, and that's the PRECISE study. And so I'll talk a little bit about that in a minute. And it's taking place at 24 clinical sites around the US. Next slide, please. So the PRECISE, let's do next slide. There we go. So the PRECISE study, the idea is that we can use immunomodulation to improve outcomes in children with sepsis-induced multi-organ dysfunction. We're targeting an enrollment of a little over 1,000 critically ill children with sepsis-induced multi-organ dysfunction. And those children will, the vast majority will be shuttled into one of 2 randomized controlled trials based on their immunophenotype, as well as 2 smaller observational studies for kids that don't meet the inclusion criteria for those studies. Next slide. So the idea behind this work is that when children develop sepsis-induced multi-organ dysfunction, they tend to fall into sort of one of 2 immunophenotypes, a hyperinflammation group or an immunoparalysis group. And so if we can target our therapies towards those immunophenotypes, our hope is that we can improve outcomes. Next slide. And so the way the PRECISE study works is that we'll have a little over 1,000 of these children with sepsis-induced multi-organ dysfunction. When they're enrolled, they'll be immunophenotyped, and approximately half will probably we would expect to fall in that inflammation group, and another half in the immunoparalysis group. For children with inflammation, they would be then randomized to either anakinra at one of several doses or placebo. And for children who have an immunoparalysis phenotype, they would be randomized to GM-CSF or placebo. And then we have, as I mentioned, those 2 smaller observational studies for sort of the extremes on either end, where they don't quite meet the inclusion-exclusion criteria for the main studies. The primary outcome is a cumulative 28-day multi-organ dysfunction score, as well as we have secondary outcomes, looking at sort of long-term outcomes, using measures like the PEDS quality of life score and the functional status score. And then we're also looking at a number of exploratory outcomes, and probably most relevant to this group, it's not really an exploratory outcome, it's more of a tertiary outcome. We're going to be looking at the anakinra PK in this population of critically ill children. Next slide. So this slide I only show just to highlight that we'll be collecting biological samples at multiple time points throughout each of these children's illness. So we have 5 biological sample collection time points over about a 2 to 3 week period. So that's going to be really important for us in terms of looking at our outcomes, to be able to look at how immunomodulation impacts these children over the course of their illness. But more for this group, one of our other objectives of this study is to create a large biorepository. And so we'll have all of these biological samples over this time period, over the course of illness, that other investigators can access. Next slide, please. And so, similar to Rachel, I think there are a number of resources and areas of collaboration between CapCorn and Imprint for others. We have currently, all the data sets from the first 3 cycles are public use data sets and are accessible by other investigators. We also have a biorepository from cycles 1 through 3, to which other investigators can access those samples. And then we'll have both public use data sets and the biorepository from cycle 4 as we complete that study. And then, similar to Rachel, we've got a robust network infrastructure that is sort of throughout the critical care units across the United States, and also has experience doing trials like this under investigational new drug applications. And I think that's all I have. So with that, I'm happy to take any questions. And I see one from Prince: is the dose uniform in the 2 RCTs? So yes. So for the immunoparalysis RCT, which is known as GRACE 2. That one, they're getting a fixed dose, a set dose of GM-CSF versus placebo. And then for the inflammation RCT, which is called the TRIPS trial, that one, they get one of 4 doses of anakinra or placebo. And it's a Bayesian adaptive randomized design. So we'll sort of, as the trial goes on, more and more children will get shunted into different groups. And second question: are we administering anakinra IV or sub-Q? We're administering it IV, mainly because in this population, the sub-Q absorption is sort of not entirely clear, because a lot of these children have severe anasarca. So when we talked to FDA about this one and went through the IND process, we got approval to administer it IV.
Thank you, Dr. Ward. Are there other questions? Questions for the whole group? Perhaps this consideration also of the early inclusion of pregnant women. This, I don't know if Dr. Nachman, if you have additional comments on that. You mentioned that those discussions actually are ongoing currently.
So I wanted to say, yes, you're right. It's horrible. I see it getting somewhat better with the antiretrovirals. And I think that's really due to the work across all the networks and the sites and the community pushing to get those studies done. I think that without the community being involved and pushing the companies, it's unlikely that they're going to change their thinking. They are so risk averse and going for the population that will bring in the most money, that it's unlikely that they're going to change their operational style and include pregnant or even postpartum women as well as children. And we fight this fight every single time with every single drug as to why would you exclude postpartum women? And they do the whole, oh, it's in breast milk. And I say, no, it's not. It's not there. Well, maybe it is, and maybe it could be. So I want to say politely, the risk averseness has changed it from people that should be involved in research to people that are being excluded from research. And as you know, we've put out several position papers in Lancet, we've gone to the FDA, you name it, we've talked about it. And I want to say politely, the landscape is only ever so slightly changing amongst the antiretrovirals, but we're not being successful amongst anything else. And the COVID vaccines are a perfect example.
There was an additional response from Kate Sticka that may be because of the concern for the fetus when studying ARVs.
So we've managed to really walk around when women are being treated with ARVs. Certainly women in sub-Saharan Africa are being exposed to ARVs on an ongoing basis. And while they were certainly thought to be excluded from the long-acting ARVs, with the incidence of pregnancy and the long-term follow-up, I think that the companies are less risk averse and are allowing it. But you're right, that first trimester seems to be an area of where no one wants to look, touch, or include, but that leaves 2 other trimesters in the whole postpartum period. So even that risk averseness about organogenesis in the first trimester isn't a good reason to exclude second, third, and follow-up. And I think that the problem is really the community is not pushing wholeheartedly. And as you can imagine with the COVID vaccines, there was that whole host of, oh, protecting my baby, but not giving not getting a vaccine, or protecting my baby and not wanting to take drug. And that resulted in our remdesivir study enrolling early, because we had so many pregnant women that did not get vaccinated, that got COVID, were admitted to the hospital, that we were able to accomplish that study in half the time that we thought we would.
It looks like Dr. Pollack included a link to, in the comments, to inclusion of a workshop on the inclusion of pregnant and lactating persons in clinical trials. That actually is June 16th and 17th, so just later this week. Aaron, did you want to make any additional comment on that, or?
It's not a new problem. I want to say everyone here, we've all been part of PRELAC. We've all been talking about it. We're taking care of patients, and I want to say we're just not moving the arrow a whole lot.
Okay, your audio is not working.
My audio is not working. Sorry if it's not.
Oh, Aaron.
Oh, I think that was me. Am I on now, Rob?
Yes.
Yeah, I mean, certainly this was a reason why Congress mandated the task force, that became called PREGLAC, to look into this. And yes, I think we would all like to have seen things move faster. The mPrint Hub itself is one thing we are doing to lower the barriers. I think as we think about how physiological-based pharmacometric modeling can be used to help reduce those safety concerns, is one way to go. The liability issue needs to be really explored. And to Dr. Walson's comment, I think there might be some question marks there about how that can apply. And that's actually one thing that one of the implementation recommendations was for the National Academy to really look into more details of the medical liability issues around this. And so that's a kickoff of this will happen at the end of this week. One other thing, there have been a recent set of FDA workshops on conducting research in pregnant and lactating people. And that is certainly, those workshops are very much directed to updating FDA guidance. So certainly there are things moving forward, and yeah, it is much slower than we would like. And I think the COVID pandemic has really highlighted, how bad it can be when we aren't conducting these studies earlier. But everybody here is, I know, trying to move the field forward.
Thank you. So are there some perhaps some ideas about how to establish some of this integration in a concrete way between mPrint and all the networks? I know actually we're already quite interdigitated, but are there other sort of elements that we might want to highlight? I don't know, Sarah, if you want to jump in and comment or try to highlight. There's certainly an amazing set of resources here.
Yeah, I think, there's others who probably can comment, have been around longer than I have. Looking at, actually, one of my thoughts is, Impact's been around since 2006 and Capricorn since 2005, and they've made a lot of progress. And I'm looking at it as a new network of, kind of in awe of what you've done. So I'm going to actually turn the question around, I think, and ask for advice from these networks on how can we be most successful. And I think working with you and utilizing some of the resources and networks you've built is a great start, but are there other suggestions that you have?
So I wanted to just mention that we have a huge database of samples and data from across our studies from multiple years. And while everyone always wants to start a new study, I want to put a comment in that we have the ability to do new work concept sheets as well as data analysis concept sheets as part of our network. And using the dashboard, being able to find the samples that are in the freezers and requesting those to be used. We do have a small amount of money that we could even help fund some of those investigations. Obviously, they have to go through the network process to be evaluated, but I want to encourage use of samples in repositories. And it's not just our repository, but NICHD has multiple repositories across the lifespan. I really think that we haven't done enough asking questions of those repositories.
Actually, that's a great segue into something I missed talking about during our KRCC meeting this morning, which is our administrative supplement that we just received. And our goal is actually to work with groups, identifying where those repositories are, what you have, not necessarily, and then providing a link to you on our website, trying to identify, where researchers and networks who have those biospecimens available for secondary analyses, and what, in some cases, what the ethical issues may be to access those or those limitations. But just making the imprint website a resource to have an access point to various biorepositories. So that's another thing. I invite everyone who's here who has those samples to send us an email or give us a comment in the chat now about support for those and how we can identify them. So, but yeah, that's a great point. And I know we'll hear from Rebecca Clifton.
The only other thing I did want to mention that I've been hearing over and over again is the lack of ethnic diversity amongst other repositories. And I want to say the good news about Impact is that our repository is quite diverse, because more than half of our sites are outside of the US. They include populations that are incredibly diverse, and between the pharmacogenomics as well as pharmacokinetics, those samples can be used not only pregnant women postpartum, but also pediatric, to look at some of those PK genomics type studies, because the samples are sitting there and they are quite diverse.
Those are very terrific points. And I think one of the projects that we're undertaking, that R21 now funded projects, basically is mining all of the publicly available resources in that area. And I think Rupsa had a question as well. I didn't want to turn her off. So,
yeah, thank you. I was just wondering, is there a plan for sort of having a system for rapidly reacting to things like COVID-19 or other conditions where, whether, even if it's not collecting biological samples, but at least collecting outcomes on pregnant patients who are receiving these medications, what trimester in pregnancy they get it. Ideally for patients who are receiving medications during labor and delivery, you even collecting cord blood to be able to document that type of exposure.
So are you asking it for the different pharma company studies, which we would not have access to, for those patients that are enrolled on our current studies, but I want to say get co-enrolled on another study, we would have that data. So it's that divide of what study they're on, do we have access to it? That's so difficult.
Oh, okay. Okay.
There were two comments also in the chat here from Dr. Walson about leveraging the political divide to find sponsors and legislators to protect the unborn child and incentivize studies in pregnant women. And Aaron shared again the notification that there will be a U24 public posted that is amenable to taking advantage of these repositories. And then Kate, yeah, and I can from Kate Stick again, my comment about the fetus was actually just the opposite. I think the reason social work has been done with ARVs because of the fetus, the concern about preventing vertical transmission. that trumps the adverse organogenesis risks. Studies for simply improving care in pregnant women aren't that valued. That's a stimulating point. So go ahead.
I'd like to say the answer to those is both is yes, we are concerned with preventing vertical transmission as much as we are concerned about preventing new infections. So I want to say all of those have contributed, but we've certainly moved forward cautiously when we're enrolling women on novel ARVs during pregnancy. So, and recognizing that women worldwide who are on prevention studies often get pregnant. So the data is always there, despite us asking them not to get pregnant.
I'd also just like to add on to what Sharon was saying. In addition to the biorepositories, all these networks have tremendous resources in terms of PK data sets. And particularly when we're thinking about physiologically based PK and we're looking for observed data with which to evaluate our models, you know, having access to the raw data would be tremendously helpful moving forward. And I think this imprint has a real opportunity here to try and help facilitate some of that.
Read the really important points, but and I think when I'm going to draw the discussion of the pharmacokinetic study networks into a close and pass the presentation over to Audrey to take the next section here. So thanks to all our speakers for really fantastic presentations and contributions.